Hello everybody, today our exhibition is about biomedical and chemical label-free sensing. So, in fact, do you know how is the label-free biomedical sensing works? It can be roughly divided into three different mechanisms: electrical, mechanical, and optical. Electrical sensors mostly based on nanowires, such as silicon, indium oxide, zinc oxide, successfully demonstrate the detection of proteins and DNA sequence based on monitoring of change in current, resistance, impedance, and so on. Mechanical transducer is a cantilever-based detection experiment. This sensing idea is detecting the change in the stress profile of the microcantilever in the specific analyte bound to the surface. On the other hand, optical sensing systems use electromagnetic wave for sensing. When the incident light shines on a metal surface, the free electrons of the metal atom will vibrate with certain frequency of the light, which generates a surface plasmon. A surface plasmon, also known as surface plasmon photon. It is an electromagnetic wave which propagates at a metal dielectric interface. That's why it is strongly dependent on the geometry of the plasmonic structure and the environmental parameters. Therefore, any change in the environment, such as the binding of biomolecules to the surface of the metal film, will change the plasmon mode. The sensitivity of SPR-based DNA and protein biosensor is very competitive compared to other existing electrochemical, electrical, and fluorescence biosensors. And you may notice that the detection limit of LSPR biosensor can be low to femtomolar. Localized surface plasmon resonance (LSPR) is our research focus, and we are confident that it will be one of the most powerful biosensing techniques with high sensitivity, excellent selectivity, quick response, and low cost. As we know that the nanotechnology has been intensive research for decades, we are now able to fabricate materials with delicate nanostructures. LSPR biosensor with plasmonic nanomaterials with large surface to volume ratio will greatly improve the sensitivity of the system. This video shows the experiment setup in our lab. This is the temperature control of the light source below. We are using a white light source to cover enough frequency of the spectrum for detection. We can increase the current to have higher intensity of light. This is the water pump for pumping the solution into the system. These black stands are polarized, which used to control the light path to the probe cell. The gas chip with stone non-ion is set up here. This is the flow cell that allows the target sample to react with the sensing surface. Here is an optical fiber receiving the reflected light. The spectrometer will record the intensity of the light during the experiment along with time. After the experiment, we can process the data to see the variation of light and analyze the result. For both biomedical and chemical sensing, we need to functionalize the material so that it can catch the target. Once the polymer captures the PP ions, the LSPR signal will be different, and then we can analyze the data to get some information about the sample. This picture shows the phase response to what we flow into the system. You can observe the large difference in the phase response between blue and red curve during the water sample reacting with our sensing surface. The one with red ions will be captured by the polymer and significantly change the surface environment. As a result, the spectrometer records the variation, and eventually we obtain a large response from the lead-contaminated water sample. Inspired by previous Hong Kong lead in drinking water incident, we have made a prototype that integrates our LSPR system into a portable device for household water quality monitoring. An advanced LSPR biomedical application could be used in cancer cell monitoring. With similar mechanism, we take one substance called exosome released from the cancer cell to capture CD147 antigen and MCT1. The concentration of two chemicals will reflect the health condition of the cancer patient. Hence, LSPR could become another method replacing biomarker for tracking the growth of tumors. Besides, one of our group members who is now working abroad just published his new research outcome. His team successfully developed a LSPR biosensor for coronavirus. We know that the current diagnosis of COVID-19 relies on RT-PCR technique, but it is very time-consuming and is likely to show false positive results. Therefore, this LSPR biosensor may provide a fast and accurate solution for clinical COVID-19 diagnosis. I want to emphasize that the LSPR detection is a powerful technique in biosensing. Researchers can develop new plasmonic nanomaterials and create various of sensing mechanisms to facilitate the detection. Recently, our team is also fabricating a normal titanium nanostructure to realize level-free biomedical sensing. I believe you are somehow impressed by our LSPR biosensors. If you are interested in this field and want to have deeper understanding, you are very welcome to discuss with us. Thank you.